Okay, let's draw some DNA. So, all right, let's talk about DNA. We're going to draw it. We're going to talk about what the parts are. And some of this information will be on the drawing quiz, and other information is necessary and may be advanced on the drawing quiz, since you don't get notes for it. We're going to talk about DNA and RNA this time. These are the molecules that carry information in your cells. The general rule of thumb, remember that phrase that we learned about, the quote-unquote central dogma. Does that sound familiar to you? That dogma is like an underlying, like a main idea or a main principle. And that central dogma is that DNA gets copied into little pieces of RNA, and then those RNA molecules are used as directions to make proteins. This is the central dogma, which once again just means that it's like a main idea in biology, something that everything seems to follow is what this means. So anyway, this is not entirely true, this central dogma, because it turns out that there's some viruses that can take, little pieces of RNA and convert them into DNA. And so a better model of the central dogma would also be to have an arrow going this way, because there are some living things that can do that backwards transcription. All right, let's talk about this word here. Nucleic acid is the kind of molecule that this is. Just like there are proteins and lipids and carbohydrates, there are also nucleic acids. And DNA and RNA are examples of nucleic acids. The start of it, ribo doesn't refer to ribosome, and deoxyribo doesn't refer to a deoxyribosome, of which there isn't one. So, what do these stand for? These are the bases, the root words of the sugars or carbohydrate molecules that are found inside of DNA and RNA. It doesn't mean that they're sweet. You don't eat a piece of DNA and think like, this is good. It's just a part of the structure. In RNA, the sugar is called ribose, which is why it's just ribonucleic acid. And in DNA, the sugar is called deoxyribose. I learned this, misspelled there. I learned this amazing thing about the difference between ribose and deoxyribose a couple summers ago. You'll have to ask me about that sometime, because it was a real neat piece of information. Anyway, deoxyribose means that the sugar molecule has one less hydroxide group, or OH group. These are both pentagon sugars, so these are both what we call pentoses. These are both pentoses, and that's going to be important, but we don't have to draw ribose with all of that detail like we did at the beginning of the school year. In fact, when we draw these sugars, we are just going to draw them as little pentagons, and that's it. Okay, so let's talk about DNA first. So, DNA is, as you know, a double helix, and we're going to draw it like a ladder. There are two parts to it. There's this whole side part here that's also found over here. We call this the backbone of the DNA. The backbone of the DNA. And this is just made of alternating sugar molecules. In DNA's case, it is deoxyribose with phosphate molecules in between. We'll add some more detail to this in a minute, but we want to have a couple of different models for ourselves so that we can understand what it is we're drawing. These are held together. These sugars and phosphates are held together with covalent bonds. They're very strong bonds. And those covalent bonds are formed using that process that we learned about at the beginning of the school year, that condensation reactions or dehydration synthesis. And you might want to flip back in your notes really quick, but a condensation reaction is when you take an OH off of one molecule and an H off of another molecule, that becomes water. And then the two things that you remove them from are able to join together to form a really strong attraction. Okay? And then we've got the steps on the ladder, and each of these steps on the ladder is a pair of nitrogenous bases. Nitrogenous bases. And that just means they're a molecule that behaves like a base, not an acid, and they're full of nitrogen. There are 4 kinds of nitrogenous bases. There is adenine, thymine, guanine, and cytosine. And you'll remember from being a human being in science classes for the last, 5 or 6 years, that these things pair up into A's and T's and G's and C's. We call these complementary pairs. And the way that these things pair up, and this is really interesting, is by hydrogen bonds. Hydrogen bonds, you'll remember, can't move up anymore. Hydrogen bonds are these attractions between a hydrogen of one atom and an oxygen of another atom, or a nitrogen of another atom, or what is the other one? A fluorine of another atom. The G's and C's know to pair up with each other because the G's and C's form 3 hydrogen bonds between the 2 of them. And the A's and T's only form 2 hydrogen bonds between the 2 of them. And that's how A knows how to pair up with T. Otherwise, you get into this unstable connection. You can't pair 2 hydrogen bonds with 3 hydrogen bonds. You have this open attraction that makes the molecule very unstable and wants to attract to other things. So, you can't do that. So, let's draw this out in a little bit of detail now. I'm going to move over here to the left. Put this wherever you need to in your notes. The drawing quiz is going to be specifically about drawing exactly what I'm about to draw right now and being able to label the different parts of it. All right. So let's start off by drawing the backbone. So the backbone, we're going to use little circles as the phosphates. I'm going to erase that, draw that just a tiny bit bigger so that I can write a P in there. This is a phosphate molecule. A phosphate is a combination of phosphorus and oxygen. Chemistry people, you'll remember that this is the formula for phosphate is PO4. Now, this is going to be covalently bonded to a deoxyribose. It's important that you attach it to this carbon right here. That's an essential part of the drawing quiz. So this is the deoxyribose on the DNA. And then we're going to move down to the right 1 carbon. And we're going to draw another bond out. You got to watch your width on this one. So make sure that this phosphate is lining up with this phosphate right here. So draw that line about that long. And then you're going to draw another phosphate. We're going to come in here and be attracted to, or we're going to be covalently bonded to another deoxyribose. We're going to have another phosphate. We are drawing this backbone right now. Here's another deoxyribose. Here's another phosphate. I'm going to come down here. And I'll draw 4 deoxyriboses. Pay attention to your drawing quiz because you're going to be told to draw a certain number of base pairs. And that number of base pairs will inform how many sugars and phosphates you draw. Okay. Now, coming off of this upper right carbon on all of these deoxyriboses is going to be a covalent bond that attaches to, and we'll just draw it like a rectangle, a nitrogenous base. Okay? And we can make them any bases we want to. I'll make this one a G, this one will be a T, we'll put another T here and a C here. You don't have to have each one represented. You just have to be able to pair them up correctly. Okay? Okay, so then these are going to be attracted to their complementary nitrogen bases. So we're going to draw another rectangle that they're attached to here. Now I'm going to draw these upside down for a really weird reason, but just stay with me here. All right, so this is a C because the complementary base of G is C. The complementary base of T is A. Another T, so the complementary base is A. And this is a C, so the complementary base is a G. Now the reason I'm doing this, the reason I'm drawing it this way is because the two DNA strands run opposite to each other. And this is known as being anti-parallel. Anti-parallel. So scientists decided to label the two ends of the DNA strands with numbers. It'd be nice if they were just 1 and 2, but they're not. The start of one DNA strand is called the 5' end, and the end of that DNA strand is called the 3' end. And so these two DNA strands run parallel to each other, but in opposite directions, which means that this end up here is the 3' end, and this end down here is the 5' end, which is kind of why we're writing it upside down. It helps you imagine that they're running opposite to each other. But because they're running upside down, it does mean that we need to draw these deoxyriboses upside down as well. So here's the easiest way to draw an upside down pentagon. You're going to make, at the end of each of these bases, you're going to make a V. And then you're going to bring a line in just slightly in towards the middle, and then cap that off with a flat line. So let's do that again right here. We're going to make a V, and then bring them in and cap that off. We're going to make a V, bring each of those in and cap it off, and then we're going to make a V, bring each of those in and cap it off. We need to attach our phosphates here. Right here is where a lot of people make a mistake, and they draw their line. Whoops, didn't mean to erase just then. That should have been on draw. They draw a line like this. But if you take a look over here, we do not have a bond, a covalent bond coming off of that top carbon. So that is not how it's supposed to be. So let's pay attention to where we are here. It is the other end of the V, what that first one comes off of. Attaches to this one right here. And we draw our phosphate. Here, here, phosphate. Here, here, phosphate. And just like how we have our 5' end ending in a phosphate up here, we need to have our 5' end ending in a phosphate down here. That's an important part of this drawing quiz as well. So these are our two strands of DNA. One strand is all over here, and this strand is all over here. So two strands of DNA running anti-parallel to each other that twist around each other in a double helix. All right, so that's the gist of it. There's a couple of other things to know. Because we have a lot of repeats here, DNA is known as a polymer. And you know that word polymer. We've talked about it before. But a polymer is a set of repeating monomers. So the polymer in this case is DNA, and a monomer in this case is called a nucleotide. So we have a lot of things repeating in DNA. We have deoxyriboses, we have phosphates, we have the bases. Even though the bases are different, there's a base on every single one of these. So what is the monomer of this polymer? It's a nucleotide. And so a nucleotide is actually three different things. A nucleotide is a set of a phosphate, a sugar, and a base. This is a nucleotide right here. So if I was to ask you how many nucleotides are in this DNA molecule, think about it for a minute, pause the video, take a guess, count some things up. The answer is there's 8 nucleotides in this particular DNA molecule. So this DNA molecule has 8 nucleotides. It has 4 base pairs. If I was to ask you to draw a DNA molecule 6 base pairs, you would add 2 more of these whole sets of pairs of nucleotides. If I was to ask you to draw it with 7, you would add 3 more. If I was to ask you to draw it with 5, you would add 1 more pair. But that's a part of the drawing quiz. The drawing quiz is going to be to just draw a DNA molecule with a certain number of base pairs and to label a bunch of different parts. A little bit of a piece of advanced information has to do with these complementary base pairs down here. The nitrogenous bases come in 2 different forms. Nitrogen bases come in 2 different forms, and those forms are purines and pyrimidines. The purines are G and A, and the pyrimidines are C and T. So you can see that each base pair is a pair of 1 purine and 1 pyrimidine. 1 purine, 1 pyrimidine, 1 purine, 1 pyrimidine. And they fit together really well because these purines are a hexagon and a pentagon together. And these pyrimidines are just 1 hexagon together. So that way the width of the DNA is always the same. All right, one last thing here. If we drew DNA, we've also got to draw RNA. RNA is real simple. Instead of being 2-stranded, it is only 1 strand. It's got ribose, which is still a pentagon. However, RNA does not, I repeat, does not have T in it. It doesn't have the thymine. Instead, it has a base called uracil, or U. So if I was to ask you to draw an RNA strand, we would draw our phosphate attached to our ribose in this case. We would draw another phosphate attached to another ribose. I drew a little bit of an extra line there. And we'll just draw 1 more here. We'll make it a small one. And then we make our 3 nitrogen bases, and let's make this one a C, this one a T. Whoops, I made a mistake there. There's no thymine. I said this should be a U. I'm a bad person. All right, it should be a U, and we'll make this one an A. And this is a little chunk of RNA right here. It's got 3 nucleotides in it, but no base pairs. All right, I think that's it. There's a lot of things that I said in here that I didn't write down. So as you were watching the video, you might want to make sure that you were writing the things down that you need to. Go back and pause this if you need to.